And I have a new recording set up today, so hopefully we are not going to have disastrous sounding things like we have had in some of the past ones, which is why they aren't posted. But if you'd like to hear what my voice sounds like covered with fun static, I can play lots of it for you. Last time we talked about the problem of trafficking, which was the first step of what kind of takes so long in making an adaptive immune response. Why it takes a while between infection and actually starting to see our adaptive immune response. So we're going to actually see a response happening starting with the material today as we look at T cells being activated. I will also tell you that some of this material is stuff that I really like, and I was having trouble dividing up what I wanted to talk about today and what I wanted to talk about on Friday. So I ended up just making one big set of slides that I'll use for both days. So if you look at the set of slides, no, I do not plan to talk about all of that today. I was just feeling like, I don't know, I couldn't give any of them up. So they all got posted together. So the first thing that we need if we're going to be having a T cell response is that we need our antigen and an antigen presenting cell present in the secondary lymphoid organ. We talked a bit last time about how free antigen can move through the lymphatics and transit into the lymph node. However, we also very frequently will see dendritic cells moving through the lymphatics carrying antigen. And so oftentimes we will see dendritic cells that are going to be a part of this process. We have dendritic cells living in many different tissues. Here we can see that there are dendritic type cells in the skin before there was an injury to the skin. And those are really immature dendritic cells. And so you'll have immature dendritic cells like the one that you see in the upper left here, living in many types of tissues. Those immature dendritic cells, which are sort of closer to being monocytes, can mature. When our cell is an immature dendritic cell, it's going to be pretty good at doing phagocytosis. You can see plus, plus, plus of phagocytosis. It does a lot of phagocytosis. It actually doesn't have a ton of MHC on its surface, and it's sort of going to be anywhere throughout the body. However, if that dendritic cell comes in contact with a PAMP on one of its PRRs, then that dendritic cell is going to change and it's going to mature. That's going to include structural changes. So here you can see a mature dendritic cell. Now it starts to have all those nice little projections that make it dendritic-y. It's also going to have way more proteins on its cell surface. So it's going to go from this nice little blobby thing with not too many proteins on its surface to this protein with lots of stuff on its surface, including way more MHC and a lot of other proteins that could be really helpful for turning on T cells. So now our cell has gone from a super good phagocytosis cell to a super good T cell stimulator cell because it has so many proteins on its cell surface. In fact, this cell is probably not going to do a whole heck of a lot of phagocytosis once it's mature. It probably did phagocytosis, caught itself some PAMP, matured, and now it's ready to stimulate T cells. And one of the biggest things that that dendritic cell will do is that it's going to start to travel. It will decide to move through the lymphatic probably using a lot of the same things like CCR7 and L-selectin that we saw last time in order to move into the T cell area of the lymph node to act with T cells. And so it's the same process that you saw before. So our immature dendritic cell will be chilling in the tissue. It might do phagocytosis. That will activate it to start making all sorts of fun and exciting new proteins that can allow for T cell signaling. And so this cell, instead of being a phagocytoser, is going to be a traveler and a T cell stimulator. T cells are also going to move into this same location based on chemokine gradients that are in the lymph node, so that in the end, we're going to get our dendritic cell and our T cell next to each other, ready to actually interact. Finally, our T cell is going to see some antigen that is being presented by that dendritic cell. And so, sort of for the next few days, we're going to kind of be talking about these aspects of T cells in the periphery. Today, we're going to see the T cell originally interacting with that antigen presenting cell to get turned on and activated. Eventually, we're going to see cytokine production. We're going to see divisions of that cell. That cell is going to change its activities. It's going to differentiate to be able to do new functions, and it's going to gain some new functions. But we're going to start with zooming in on this MHC peptide T cell receptor interaction for right now. So in some ways, this is the activation of T cells and kind of what we're going to spend the next few days talking about in some different parts. This is also a little bit of an oversimplification. So one of the real things I want to just tell you briefly right now about activating T cells is that in reality, T cell activation requires 3 signals. They are called signal 1, signal 2, and signal 3. Today, we are going to be talking about signal 1. Maybe if I have a little time, I might say a couple things about signal 2 at the end, but signal 2 is really going to be Friday's deal. Today's really about signal 1. We'll deal with signal 3 a little bit more on Friday and then on Monday. And so first thing just that you want to know is that everything we're talking about today is largely part of signal 1, but there are some other signals that are in fact important for this process as well. I will also tell you that this information is still actively being studied by many immunologists. So just to give you an example, this is a paper that came out a week ago examining these exact issues. And I just want to show you 2 figures from this paper. So this is a description of what they were saying a week ago this process looks like. That looks super fun, right? And this is what they say, if they only look at what happens in the first 10 minutes of the process, this is what they come up with. So as much as you may look at what I tell you about today and say, oh my gosh, she's telling me about so many proteins, I am really simplifying compared to what we actually know. And every protein I'm telling you about, I'm picking for a reason and telling you about for a specific reason. So again, realize that we are taking quite a lot and sort of boiling it down to a few activities that are happening sort of as this pink arrow of signal 1 today. In our discussion of signal 1, our T cell has a goal. So when we are activating this T cell, there is a goal in this activation. Yes, signals 2 and 3 are part of this goal too. But for today, you can think of we're signal 1 and we got a goal. And that goal is that our cell wants to make this cytokine. And this cytokine is called IL-2. So really, to get a T cell that is fully activated and is going to do a lot of good stuff, we need our T cell to make IL-2. And so our T cell needs to make some changes in terms of its transcription in order to transcribe IL-2. And IL-2 is super well studied in immunology. And so what we know is that in order to transcribe IL-2, we need to have 3 transcription factors made, and they need to be all made at the same time. We need a transcription factor called NFAT, we need a transcription factor called AP1, and we need a transcription factor called NF-kappa B. And if those 3 transcription factors are made, are all 3 made, sort of are made in combination, they can all bind in this promoter region and lead to IL-2 transcription. So with all of the signaling we're going to talk about today, all of it is going to be either to turn on NFAT, to turn on AP1, or to turn on NF-kappa B. Because it's all leading us to this goal of making IL-2. This starts with signaling from the T cell receptor. Yeah, Mark. Okay. This starts from signaling with the T cell receptor, like I have told you about before. So you're going to see a couple of pieces of this that are similar to something I've told you before. I remember that I told you that we have this co-receptor, CD4, in the case of a CD4 T cell, that will bind, in this case, to MHC class 2 to help stabilize the interaction between our T cell and our antigen-presenting cell. In reality, we also have adhesion molecules that are holding the cells together. Specifically, we have integrins on either side of the T cell receptor that are actually giving us a really strong hold between that T cell receptor and the antigen-presenting cell so that we can get a long-term contact and long-term signaling. There has been a lot of work done to characterize all of these proteins that are on the surface of both the T cell and the antigen-presenting cell. So you can see we've got the MHC on the antigen-presenting cell, the T cell receptor and CD3 on the T cell, CD8 or CD4 on the T cell. We've got this second set of signals, this CD28 with CD80 or 86, that's signal 2, which we'll deal with at some point. And then you can see that we've got these integrins on the sides that are almost making a wall, that are sort of keeping all of this material kind of together for signaling. People have spent a lot of time studying all of these things, and they can actually look at the way these proteins are assembled on the cell in a microscope, and they refer to this region as the immunological synapse. Sometimes there are arguments between immunologists and neuroscientists about like whose thing is more complicated, and they're all dumb. But immunologists have started to take the word synapse, and we use it in all sorts of things. Technically, a synapse is just a junction between 2 cells. And so this is the immunological synapse, where we have the T cell with the antigen-presenting cell. And if you actually do localization, you can see that all of the proteins make almost like a target in their organization. You've got this central area, officially known as the CSMAC, or the central supramolecular adhesion complex, that includes the T cell receptor, the co-receptor, and the things that are involved in signal 2. And you have this outer ring that has all of your adhesion molecules sort of holding all of this together. And so people have been able to actually image the organization of all of these proteins on the surface of the cell pretty nicely. This also is pretty cool in another way. When we put our T cell receptor and our T cell and our antigen-presenting cell together, you can see that in the area where the 2 are close together, they're close together. And sorry, I got halfway through that sentence and I'm like, this is bad. But you can also see at the edges, because of sort of the way they're curved, there's more space, whereas here there's not so much space where they're close together. what ends up happening is that there are some proteins that have super big extracellular domains. And the most famous one is CD45RA that gets shoved out of the way. They can't fit in there. They're too big, so they get pushed out. It turns out CD45RA has this cytoplasmic region that includes a phosphatase that removes phosphates from stuff. It's actually a negative signaling protein. And so when the T cell and the antigen presenting cell come together, you shove away the negative protein so that you can do lots of activation. The negative protein is just too big, it doesn't fit, so it gets shoved out of the way so that all of the tiny proteins can bind together for signaling. So I always think that's pretty cool. So in our first step of this process, we see something that I have told you a bit about before. We have our T cell receptor that has very little cytoplasmic domain. Fortunately, it travels around with its friends, the CD3 complex. The CD3 complex contains these 6 different proteins, each of which has one of these little yellow boxes, which is an ITAM, an immunotyrosine-based activation motif that can get phosphorylated if there happens to be a kinase around. One thing I will just tell you is that often, is that tyrosine, the one-letter abbreviation for tyrosine is Y. And in fact, all of these have the amino acid sequence YXXL in the ITAM. Either CD4 or CD8 is associated with some type of kinase. The most famous of these kinases is the one called LCK, which is technically with CD4, but whatever. These kinases are members of something called the Sark family of kinases. And so here you're supposed to imagine CD4 is far away, and here it's close. I know that the distance between these two figures is not particularly different, but this distance is far, and this is close. And you can see that when MHC plus peptide comes around, we can see T cell receptor binding, and CD4 is also brought into proximity of the T cell receptor and the CD3. That brings LCK, our kinase, in proximity to the cytoplasmic domains of CD3, and it allows LCK to phosphorylate all of the ITAMs. And so you can see that our ITAMs are now getting these pink dots, which are the phosphate groups, which they did not have previously. So our ITAMs are getting phosphorylated by LCK. Officially, what you're seeing here is known as receptor proximal signaling, which really just means it's near the top part of the pathway. It's close to the receptor instead of close to the bottom part of the pathway. If you remember, I told you guys at one point about a conversation I had with one of my roommates in college about immunology, who is now a district attorney in southern New Jersey. And she is not a science person. And I was explaining to her that immunology is really all about shapes and matching shapes. It's actually technically just, advanced kindergarten. And this is one really important place where we can think about shapes and matching shapes. When we have added phosphate groups onto all of these tyrosines, we have made a new charge and a new shape on these proteins. And so now there is a new binding surface that was not there before because of both the charge and the shape that we can see on the ITAMs. And because we have this new binding surface, a new thing can come bind. And so the next thing that will bind in this situation is a protein that's called ZAP70. So we've got ZAP70. In reality, ZAP70 is also a kinase. And some of these are phosphorylated by LCK, some of them are phosphorylated by ZAP70. And you can see ZAP70 has this little area where it can bind nicely to phosphorylated tyrosines. And it can phosphorylate anything else that needs phosphorylating, ZAP70's got it. Is basically the easiest way you can imagine this. And so right now, you can see that our signaling has all been based on just putting things close to each other that hadn't previously been close to each other, binding things that couldn't previously bind because we made a new shape. And all of that signaling has happened in sort of the language of phosphorylation, the language of kinases. Moving on here. At the next step, we stop having some kinases. We move to a different kind of protein. The two famous proteins that we see next here are called SLP76 and LAT. I don't remember what SLP76 stands for. Something. Something like protein 76. LAT is the linker and activated T cells. I know LAT's the linker and activated T cells. Both SLP76, LAT, and one that is shown on one of the later figures that's called GRAB2. All three of those proteins are in a different class. They are not kinases. They are all a type of protein that is called an adaptor protein. So, Mark, you went to South Africa this past winter break. As it is true. And when we were in South Africa, we needed an adapter. Right? What does the adapter do?
It allows us to plug our plugs into their outlets by giving it a connection.
Yeah, so basically we plug it into their outlets because they have weird outlets, and then I can plug in my phone. So it basically just switches the type of signal. It switches it from South Africa weird 3-prongy thing to US 2-prongy thing. Yes, M type to B type, but whatever. Did it have any other functions? Did it like change the voltage or do any, or like, I don't know, sing a song or anything? No, it did absolutely nothing except sort of just sit there and allow you to change things, right? That's kind of what adapter proteins do too. They have no enzymatic function of their own. They are largely binding sites where other things can come together. Every so often you'll hear people call them scaffold proteins, where they're just proteins where a whole bunch of other proteins can come and bind and assemble. That's why I point out that LAT is called the linker for activated T cells. It's basically just linking a bunch of other proteins together, acting as a linker protein. And so at this point, we have our adapters. And so here it's called a scaffold, but it's the same basic idea. And so we have these adapters. They can get phosphorylated, and then they just have tons of binding sites. And all sorts of proteins can bind, like you see here. This orange one, and this yellow one, and this blue one, and this purple one, and this yellow one. And suddenly you can get all these proteins in proximity and allow them to start working together where they never would have been in proximity before. The adapter is doing nothing other than sitting there and gathering all of these proteins. And so after we have ZAP70 activated, ZAP70 is able to phosphorylate our adapters and also allow our adapters to be recruited because they can bind to some of these new sites that ZAP70 has created. When we get to the adapter step, I was getting ready to say things get complicated, but I don't know that it's actually that complicated. When we get to the adapter step, we get branches. So multiple different things can happen after the adapter. We can have a orange pathway, and a blue pathway, and a purple pathway, all coming off of this adapter. And so one thing that you might notice or might think about is that we're going to start to go down branches at this point. So I'm going to go like branch, there's going to be 3 of them. We're going to go branch 1, and then we're going to come back up to lat, and we're going to go branch 2, then we're going to come back up to lat and go branch 3. One thing to notice is if you got rid of, say, LCK, or if you got rid of ZAP70, you'd kill all 3 branches. If you get rid of some of the proteins I'm going to tell you about with these branches, you'd only get rid of that branch and the other ones would still be okay. And so that's another important thing to think about. Officially, when something is receptor proximal, that means you get rid of all the branches. Is usually the way that we talk about that. So I'm going to go to the slide for the first branch that I'm going to talk through. But then I'm going to spend some time talking about it. So don't get too hung up on all the details of the slide. We're going to get to those details. Right now I'm just going to go there, and I'm going to tell you the name of the branch. And then we're going to talk about the branch. And by the time I'm done talking about it, if you remember the name, you know how everything works. So the first branch that happens downstream of our adapter proteins is the Ras-MAPK pathway. I am going to explain the Ras-MAPK pathway the way that it was explained to me in my intro cell bio class, which is sort of like a backwards kind of way, but it has stuck with me and I've known this pathway backwards and forwards ever since. And so when I learned the Ras-MAPK pathway, I learned it from the bottom. One of the big things that happens at the end of the Ras-MAPK pathway is the cell might start to divide. So division often is part of this. There's a fancy word in biology for division. What's the fancy word? Mitosis. And so what you might imagine is that the Ras-MAPK pathway involves turning on some proteins that are involved in mitosis. You might even call them mitosis associated proteins. FYI, there is actually no mitosis associated protein. Like, there are tons of them, but there's nothing called mitosis. There's not like one that's actually called this. I'm just using this as a general name of proteins. If you decided you were going to make an abbreviation for a mitosis-associated protein, what would you call it? Again, there is in reality no actual protein called MAP, but it helps us think this through. So these MAP proteins, the proteins that function here, it turns out they all are regulated by phosphorylation. They all get turned on by phosphorylation. If you were in charge of the world, what would you call the thing that phosphorylates MAP? You would call it MAP kinase. We call it MAP kinase or MAPK. There are a group of proteins that act as MAP kinases. It turns out all of the proteins in that group are regulated by phosphorylation. They only get turned on if they get phosphorylated. If you were in charge of the world, what would you call something that phosphorylates a MAP kinase? What would you call it? I would call it a MAP kinase kinase. Turns out there are a group of proteins that function as MAP kinase kinases. All proteins that function as MAP kinase kinases are regulated by phosphorylation. They must be phosphorylated by something else in order to be turned on. If you were in charge of the world, what would you call a protein that activates a MAP kinase? False. What would you call something that turns on a MAP kinase? A MAP kinase. There are a group of proteins called the MAPKKs, or the MAP kinases. If you were in charge of the world and you wanted to turn on a MAP kinase, you would use a protein known as Ras. And so if you remember that one of the things that gets turned on downstream of LAT is the Ras-MAPK pathway, you basically know all the members. So Ras is called Ras. There are a group of proteins called MAP kinases, triple, double, single. There are specific examples. There is a common MAP kinase that is used here. There's a common kinase. There's a common kinase. So I'm going to give you specific names. But this overall structure is the overall structure that always happens in a MAPK pathway. So in the case of T cell signaling, the MAPKK is called RAF. The MAPKK is called MEK. The MAPK is called ERK. And the MAP is called FOS. And so LAT activation will turn on the activation of RAS. That will turn on RAF. RAF phosphorylates MEK. MEK phosphorylates ERK. ERK phosphorylates FOS. And so this is sort of what happens in that pathway. Now, if you remember back to the beginning, I told you that there was a goal for our T cell. Our goal of our T cell was to make IL-2, and we needed 3 transcription factors. One of them was called NF-kappa-B, one was called NFAT, and one was called AP1. We don't have anything that sounds like any of those with this. It could. But what actually happens is that there are multiple versions of this pathway that get activated here. One MAP kinase we turn on is called ERK, and ERK turns on FOS. Because of other stuff, we also turn on a MAP kinase called June. No, that's wrong. We turn on a MAP kinase called JUNK. JUNK turns on June. JUNK is June kinase. So we turn on a MAP kinase called JUNK, it turns on June. We turn on a MAP kinase called ERK, it turns on FOS. These 2 proteins, when bound together, are AP1. So in the end, we get AP1 activation because we made a FOS and we made a June, and so together they're AP1. And then we get 1/3 of our transcription process happening. So that's the Ras-MAPK branch. We can go back up to LAT, and we can think about the second branch. The second branch includes a protein called PLC gamma. So LAT turns on this protein, PLC gamma. Now, in order to understand what happens with PLC gamma, it might be helpful for you to know what PLC stands for. PLC stands for Phospholipase C. So PLC gamma is Phospholipase C gamma. So I told you this is called phospholipase C gamma. It is, in fact, an enzyme, and it has a job. What might you guess the job of phospholipase C gamma is? What do you think, Rob? Not quite. Yeah. Yeah.
Break off the phosphate head from the lipid.
That's a little specific. It's just to break a phospholipid. It's to cleave a phospholipid somehow. A phospholipase cleaves a phospholipid. In this particular case, it is cleaving the head off, but in general, a phospholipid or a phospholipase just cleaves a phospholipid. Phospholipase C gamma specifically acts on this phospholipid, PI 4,5 bisphosphate, known to cell biologists everywhere as PIP2. So PLC gamma cleaves PIP2. PIP2 is just hanging out in the membrane. It's got these, see this gray box? That's the membrane. And it's just chilling. It's just present there, the membrane. When PLC is around, it cleaves PIP2 into two different products. One of them has the long fatty acid tails. It stays at the membrane. It's called diacylglycerol, or DAG. The other one is basically this sugar head. It's called inositol 1,4,5 triphosphate, again known to all cell biologists as IP3. This is the job of phospholipase C gamma. And remember how I told you that there are three branches here? One branch is what happens with DAG, and the other branch is what happens with IP3. So actually this is like 2 and 3, all smushed together. So that was number 1. This is 2 and 3. It's just a question of whether we follow DAG or whether we follow IP3. IP3 is sort of the famous one. Immunologists, we know us all about some IP3. Honestly, I'm going to tell you about DAG kind of because I always felt bad for DAG, because it would all just be left there and no one would ever talk about it again. So I'm going to tell you about DAG because I wanted to not be left out. Yeah, Paula?
Is there like a preference between DAG and IP3? Meaning, like, is there one more key than?
No, so it cleaves this, and it cleaves it right in half. So one of this makes one of each of these. So we're first going to follow IP3 and see what happens with IP3. So PLC gamma, you can see it is binding to our activated adapters. PLC gamma will go up to the membrane. It will cleave PIP2, and one of the products will be IP3, that's small sugar. IP3 is going to travel to the ER. And it is going to bind to a specific protein on the ER membrane. If you were going to name the protein that it bound, what might you name it? IP3 binding protein. What else could you name it? Huh? ERP, okay. You're overthinking this. How about IP3 receptor? So in fact, IP3 binds to the IP3 receptor on the ER. And this leads to an important change. The cell normally works super hard to keep calcium out of the cytoplasm. So normally there is no calcium in the cytoplasm. You can see that there's calcium outside the cell, aka blue dots. You can see that there's calcium in the ER. There's also a little calcium in the mitochondria, but there is none in the cytoplasm. When IP3 binds to the IP3 receptor, that allows calcium to flood out of the ER, and suddenly we get a lot of calcium in the cytoplasm. In full disclosure, we actually get some stuff happening at the membrane too, and some comes in that way as well. but we don't really need to worry about that. But you can see IP3 binds to the IP3 receptor, and out comes all the calcium. And suddenly, the cytoplasm is full of calcium, even though it was not before. This is a super common signaling thing. For those of you who've taken neuro classes, neurons do this all the time. And this is sort of generally a second messenger system because you can have one IP3 leading to hundreds or thousands of calcium ions. And so you can really amplify the size of your signal. In the cytoplasm, we have a protein called Calmodulin. Calmodulin will be able to bind to this calcium. The calcium was not previously there, so Calmodulin was previously lonely. Now calcium's around, Calmodulin will bind to those calcium ions. That will make a conformational change in Calmodulin so that Calmodulin can do a thing. If you took a lot of neuroscience, you would have also seen Calmodulin. And so that's why I said Calmodulin does a thing, because it does one thing in neurons and it does another thing in immune cells. But in any case, Calmodulin binds the calcium and does a thing. In immune cells, we see Calmodulin undergo a conformational change. It turns into a wrench. I don't know. That allows it to bind another protein called calcineurin. And calcineurin can bind to a final protein called NFAT. NFAT usually is covered in phosphates. And as a result, NFAT is not allowed in the nucleus. NFAT would love to go to the nucleus, but it cannot. The phosphates block its entry into the nucleus. If, however, calcineurin is active, calcineurin removes the phosphates from NFAT, and now NFAT can go to the nucleus and start transcription. And so this is our second of our 3 transcription factors, NFAT. NFAT officially is the nuclear factor for activated T cells. It comes from this calcium signaling pathway. Yep, Mark.
Does NFAT have cytoplasmic function?
Not to my knowledge. I don't think so. As I mentioned to you guys before, this is one of those pathways that every immunologist knows. This is one that we all got down. And one of the reasons why this is such a famous pathway in immunology is that one of the first really famous drugs that alters the immune system was a drug called Cyclosporin A. This was the drug that allowed transplantation to be possible and is a drug that allows immunosuppression in a number of different clinical conditions. Cyclosporin A specifically blocks Calmodulin-calcineurin interaction, and so we can have calcium signaling, but NFAT can never be activated. NFAT can never go into the nucleus to result in transcription. And so this drug has sort of revolutionized and was sort of the first type of immunotherapy that was ever possible. It specifically acts on this pathway by blocking the calcineurin-calmodulin interaction. So cyclosporin is this orange pill thing. And now NFAT stays permanently inactive, keeps its phosphates, chills in the cytoplasm, even though there's calcium, and we never see transcription. And so our T cells never get activated because they can't have their goal of having 3 transcription factors. One of the 3 is blocked. Yes?
Would that work in all T cells?
It would.
So, like, isn't that really bad?
So there are some problems with cyclosporin, but multiple therapies were not possible. There are multiple clinical, transplantation, for example, was not a thing until cyclosporin, or was not a frequent thing. But yeah, there are clearly problems with it. So this is our second pathway. We can then also think about our third pathway. And our third pathway comes from DAG. Remember, we left DAG when we had PLC gamma, we cleaved PIP2, we made DAG, diacylglycerol. Really, it's in the membrane. I don't know why they drew it down here, other than because it would be hard to make a messy drawing. DAG leads to a number of different signaling events that eventually lead to NF-kappa-B activation. So DAG actually ends up turning on NF-kappa-B activation. I'm not going to go through all the details of getting from DAG to kappa B because there are about 19 convoluted ways. But the moral of the story is DAG turns on NF-kappa B. And so that's how we get our third transcription factor. That's how we have accomplished our goal in our T cell in order to activate our T cell. And so in the end, this is the process. We have our receptor proximal signaling cascade, where our T cell receptor binds to MHC plus peptide along with CD4 or CD8. That brings in the kinase LCK to phosphorylate ITAMs. That allows ZAP70 to come in and bind and also phosphorylate. That will bring in our adapters, GRAB2, LAT, and SLP76. That will activate RAS and the Ras-MAPK kinase pathway to turn on AP1. We'll also turn on PLC gamma, which will cleave PIP2 into IP3 and DAG. IP3 will turn on the calcium pathway and make NFAT. PIP2 will make DAG, which will turn on NF-kappa-B. And when those three things have their powers combined, they're IL-2 and not Captain Planet. And so we can put together those three transcription factors and make our active T cell. So I'm going to talk about this next part sort of partially. I'm going to do some of this, then we'll come back to it again next time. But that IL-2 is really important for our T cells in the periphery. When IL-2 was first described, it was not called IL-2. It was actually called TCGF, T cell growth factor, because T cells need it to grow. And so what ends up happening is that these T cells make IL-2 that signals back on them in autocrine to help them grow, as well as signaling to their neighbors in paracrine. And so all the T cells make IL-2, and that helps all the T cells grow, and everybody's happy. And that also allows for replication and multiple cell cycle progressions for those T cells. Everything I have just told you about, well, not exactly the IL-2 part, is really part of the signal 1 signaling. But in order for our T cell to be properly activated, that T cell requires a second signal, signal 2. The cytokines are signal 3, and so IL-2 kind of is a cytokine. But signal 2 is a critical part of this whole process. And in fact, there might be some issues that you had with T cells earlier in the semester, and signal 2 solves a lot of them. Signal 2 is pretty great. So here you can see this in another view, that our T cell activation requires two signals. One of them is the signal that we just talked through. The other one goes through some co-receptor proteins. They will lead to signals that may sort of add onto or tweak what I've just told you, though they don't actually it's really its own signaling cascade. It's not related. And so that's why these are referred to as the co-receptor. This is a co-stimulator. It sort of like spices up signal 1. It doesn't actually completely alter signal 1. For signal 2, our T cell has a protein known as CD28. And our antigen presenting cell has either CD80 or CD86. CD80 and CD86 have other names as well. CD80 is also called B71. CD86 is called B72. And so sometimes you will hear people, and you will hear me, just say B7. And when we say B7, we mean either 80 or 86. It's fine. It's basically the co-stim guy over here. Because it was first described as B7, and then we're like, okay, also CD80. And then someone was like, oh, actually, there's two. And so it was originally described as B7. Most immunologists learned it as B7. We now know there are two different versions of it, CD80 or 86, also known as B71 or B72. And so in order to activate our T cell well, we need to have that T cell receiving a signal from signal 1 from MHC plus peptide, as well as from its co-stimulatory molecule B7. You can understand how the antigen presenting cell knows to make MHC plus peptide. They just present on MHC plus peptide. If it's class 1, these cells are always presenting on class 1, all the time. Self antigens, non-self antigens, it's just a part of a cell's job. So they don't have to be told, make class 1, they're just doing this all the time. With class 2, we do see some initiation, some activation of class 2, but our professional APCs present class 2 pretty often. So you might wonder, well, how the heck does the cell know it's time to make the B7? That it wants to make B7, it wants to turn on a T cell well. And the way that cell knows how to turn on co-stimulatory molecules like B7 is as a result of PRR signaling. This was originally described as the danger signaling molecule. A lot of immunologists have gone away from that. For a while, when I was an undergrad in particular, that was like, people laughed at that model. And so it's one of two things that I teach you guys that sometimes I say it, the danger signaling molecule, and I expect like lightning to strike me down. So this is the danger model. And the idea is that our cell is going to be presenting on MHC kind of all the time. But it's not always going to be making signal 2. It only is going to make signal 2 if there happen to be some PAMP or DAMP around in order to say, hey, that antigen that was on my MHC is bad. We got to make a response, guys. Here's signal 2. Make the response, please. Something dangerous is around. And if there's no PRR stimulus, if there's no PAMP, then we never make signal 2, and we don't tell the T cell, hey, it's dangerous, you better do something. And so you can imagine this deals with a lot of that presentation of self-peptides. Many of the self-peptides that are being presented by your cells right now are being presented without any danger. There's no PRR stim by that same cell. And so the T cell is not going to get fully activated. It's not going to get signal 1 and signal 2. It's only going to get signal 1. And so this is sort of a nice way that we can integrate this and allow for the requirement of some sort of outside signal in order to turn on this process. Yep?
You answered the first part of the question.
Okay.
The second part is, how is that evolutionarily feasible for energy conservation to have the first signal happen and then the second one not happen? Why?
So it's feasible for a couple reasons. The idea is that by having a signal 1 be sort of happening easily, then you get breadth. But you control that breadth with signal 2. And this is, in fact, one of our examples of peripheral tolerance. I go through peripheral tolerance later, and we go through like 6 examples, and this is actually one of them, spoiler alert. So this is sort of a key thing. But the other piece to this that sort of also talks about an answer to Lexi's question is that typically our T cell will see signal 1 and signal 2 and will become activated. However, if a T cell gets signal 1 without signal 2, that is an indication that that's probably a self-antigen. And so in fact, not only does the T cell not get activated, the T cell is energized. So with B cells, we saw B cells becoming energized in the bone marrow. T cells become energized in the periphery when they see signal 1 without signal 2. And so this can be a nice way to determine whether or not something is foreign. And so there's some really interesting ways that this is sort of evolutionarily important. In addition, some of you may have heard about checkpoint inhibition cancer therapy, which won the Nobel Prize in 2018 for medicine. It involves playing with CD28. It's actually all modifications of CD28. And so we can talk a little bit about how that works with CD28 as we go forward. So this is an incredibly important and just won the Nobel Prize pathway that we're talking about here.